Go. Can everybody hear me?
Yep, sure can.
All right, great.
All right, so next up here we have Megan Tomamichel. She is interested in host parasite dynamics in aquatic systems. Her previous work includes modeling the consequences of a microsporidian parasite in yellow perch populations, exploring trophic cascades in freshwater ecosystems, and researching functional genetic mutations in an invasive freshwater plankton. She is working on her PhD right now at the University of Georgia and is co-advised by Dr. Richard Hall and Jeb Byers in the IDEAS program. So thank you very much, Megan, for joining us today, and you can take it from here.
Great, thanks, Nick. I'm here to talk today about some of my ongoing research on shrimp blackgill, which is a disease that may be threatening Georgia's food shrimp industry. A quick outline, I want to introduce blackgill, what it is and what causes it, and then importantly, like when it is. I want to talk about some experiments that are currently underway, and it would be great to get people's feedback about those. And then finally address like what evidence we have that shrimp blackgill is causing decline in the industry, and some management applications or potential management strategies, including fishing out the parasite. So this is a diagram from a recent paper, Landers et al. 2020. And it describes what we know or what we hypothesize is the shrimp blackgill life cycle. We know that it's caused by a recently described ciliate called Hyalophysa linei. It has a histotrophic life cycle where free-swimming tomites attach to shrimp gills and then replicate using the nutrition that they gain from feeding on the shrimp gill tissue. They produce tomites while on the gills, and then eventually those tomites like burst into the free-swimming infectious stage. We also think that there's a potential exobiotrophic life cycle where instead of feeding on the shrimp gill tissue, they feed like harmlessly on the shed exoskeleton, but that hasn't been confirmed yet. We also believe that shrimp have the potential to shed the infection with their molting cycle through the exoskeleton, because shrimp actually do shed gill tissue. So the disease is very striking because once the swimming tomite latches on to shrimp gill tissue, the shrimp's immune response produces these like melanizing compounds that turns the gills black. Here's a microscope image of what that looks like. So the arrows are pointing out the ciliates, and you can see over on the right side, there's like a dark cloud of like the melanizing tissue. It's unclear if the shrimp blackgill will kill the shrimp outright, but it definitely does reduce their ability to sequester oxygen from the water. And this is especially problematic when the water temperatures get really warm and there's less oxygen available. We also have some evidence that it reduces the shrimp's ability to escape predation, which is not good for all the shrimp that are out there. The shrimp blackgill, there definitely does appear to be this distinct cycle. Over on the left, you see a graph with the shrimp blackgill prevalence, and the bars are the visual blackgill, so shrimp that are coming into our lab, and look, we can see that like black gills on them. The dots in the line is the PCR blackgill prevalence, and you can see that there's evidence that we have like asymptomatic shrimp. And so on the x-axis are the months of the year, and there's a distinct peak into the late summer, fall season, which could be described by water temperature, or could be caused by water temperature. In the fall is actually when the coastal Georgia waters are the warmest. However, this peak also could be because of the nature of the shrimp life cycle. In the spring, shrimp return to spawn, and then The baby shrimp spend about a year out in the ocean, like as plankton, and then they're swept into the coastal and estuary waters, where they mature into subadults. And at this point, that's when they start marching out towards the estuary, and this occurs at the same time as the peaks in blackgill, so like late summer, early fall. And looking at the prevalence graph again, it seems like there isn't a lot of shrimp blackgill prevalence like in the other seasons of the year when shrimp are captured. So it could be that shrimp are actually bringing the infection from the estuary out with them. Or it could be because the densities of shrimp near the coastal waters are exaggerating the transmission of the disease. So I have two primary experimental questions that I want to address over the course of my PhD. The first is waterborne transmission a significant source of infection? So basically, are there a lot of these free-swimming tomites out in the ocean infecting shrimp? And if it's the case that this is a significant source of infection, does this rate of infection change because of the season, the different habitats that shrimp are occupying over the course of their life cycle, or the temperature? I also want to try and detect if direct transmission, so shrimp that are in close proximity to each other, not necessarily just like oceanic water, cause a significant source of infection. And if so, does this rate of infection change because of the density or the frequency of encountering infected shrimp? And I'll talk a little bit more about what I mean by that further on in the presentation. So step one is that we have to cure the shrimp. We do not have access to shrimp that have not been exposed to the ciliate. So we've developed a method of purchasing shrimp from local bake shops and then administering an over-the-counter aquarium medication that's been designed to combat Ichthyophonus hofferi. So we administer the drugs with a 50% water change every other day, and the shrimp are held in artificial seawater to make sure that they're not being like re-exposed to any potential tomites that are in untreated estuary water. After 2 weeks, I sacrifice a representative sample of our cured population and test their gill tissue using PCR. Unfortunately, the test involves sacrificing the animals, so we have to like grab a certain number to give us an idea of what the prevalence looks like overall in the cured population. And the most recent results from this is that we had an 80% infection rate and then down to 10% infection rate after the 2 weeks of curing. So the first experiment that I've done this summer is just trying to address whether waterborne transmission actually is a source of infection. So we've built what I call shrimp deployment units, or SDUs. And over on the left is a picture of it. They hang off of the floating docks in the estuary, and each of the black cages houses one cured shrimp. Recently, I deployed 45 cured shrimp in this manner over a period of 9 days, and then sacrificed all the shrimp and checked them for infection using PCR. Here are results from that. So the first row are all of the deployed shrimp, and they do test positive using our genetic test. The bottom row are control shrimp, where I put them individually in cages and then just house them in instant ocean over the same time period to make sure that we didn't have any like latent infection cropping up. And then the last 2 bars are the positive and negative control. After finishing all of the genetic testing, I found that there's an 83% prevalence of shrimp blackgill over the course of 9 days. I went back to confirm under the microscope a subset of these infections. And here's a photo that I took on my phone of that like microscope slide, the squash slide. You can see that at the end of the shrimp gill tissue laminae, there are these like circular kind of grape looking things hanging on the end of it, and those are the ciliate, and it's highlighted by those gray arrows. You can also see like the line of melanizing molecules that are marching towards the ciliate in response. So to further explore waterborne transmission, I want to test whether this transmission rate varies across seasons and across the habitat. So the first one I did was the estuary, which I just talked about. The estuary, I'm defining it as like a water body that large ships can go through, such as our research vessel Savannah pictured here. And the habitat for the estuary is characterized by being like pretty close to oceanic salinity and also pretty stable water conditions. And the biological community is characterized by being surrounded by a lot of grass shrimp, but less contact with like other crustacea, such as like fiddler crabs and things like that may be harboring the parasite. Now I want to expand to look at the marsh and ocean influenced habitats. The marsh is characterized by being surrounded by lots of different species of crustacea. It has lower salinity, but high temperatures and lower oxygen, so it may be more stressful for the shrimp. And then I also want to look at the ocean, which is actually where the shrimp are harvested, to see if there is any waterborne transmission happening out there. And ocean habitats are defined as like not that much contact with a lot of other crustacea, but very stable water conditions with full saline water. The final element to this waterborne transmission question is going to be untangling any seasonal changes. I forgot to mention that I wanted to do each of those 3 habitats once a season. So I want to untangle any like seasonal changes between that and like whether the temperature is driving those changes. So I'll take like untreated seawater pumped in from the estuary and either heat or cool that water, as well as having an ambient experimental unit, and then measuring the transmission between different batches of cured shrimp. And again, I'll try and do this at similar times as our seasonal shrimp deployment unit experiments, to really try and untangle like whether the differences are because of temperature or some other factors that vary seasonally. So on to like the second set of questions. Density versus frequency dependent transmission is a disease ecology term that reflects whether or not transmission increases with population density. I've taken this graph from the excellent Parasite Ecology blog, and the solid orange line represents density dependent transmission, where as population density increases, the contact rate increases and you get more transmission. These are diseases such as COVID-19, where they're often spread by infectious particles in the air, and the more susceptible individuals that are exposed to the parasite, that means the more of them are going to get infected. The dashed line, it represents the frequency dependent transmission, and you can see that like it eventually levels out as far as the transmission goes over the population density axis. So no matter how many susceptible individuals you cram into the area, there's not going to be a corresponding increase in transmission. A lot of these diseases are thought of as like sexually transmitted diseases, where it doesn't really matter how many individuals are in the room, it's the encounter rate with a like infected individual. So here is a design that I'm working on to try and test whether the shrimp blackgill infection increases with density or the frequency of encountering an infected individual. Over the horizontal axis is increasing density of shrimp, and then the vertical axis increases the proportion of individuals that have been exposed to H. lining. So, and also these are just like representative images. I'm really hoping to have more than like 4 shrimp in the 1-by-1 treatment, but we'll see how that goes. So yes, I'll be able to determine whether it's more likely that H. lining increases with the density if there are changes along that horizontal axis, or whether it's a more frequency dependent disease if there's changes between the vertical axis, whether like we see more transmission when half of the shrimp are already infected versus just increasing the density of shrimp. So it's very important to try and determine the nature of transmission of this disease, because it limits our management options depending on how the disease is transmitted. There's this concept of fishing out the parasite, which basically implies that if it's a density dependent, directly transmitted pathogen, we may be able to fish the shrimp population low enough to actually halt transmission altogether. So that would depend on if we can fish the shrimp density low enough on key times of the year to stop this potentially directly transmitted density-dependent pathogen. However, if shrimp blackgill is primarily transmitted through waterborne transmission, our option is probably going to be to fish around the parasite. So basically advising shrimpers to fish at certain times or certain temperatures to prevent them from acquiring large catches of infected shrimp. So is shrimp blackgill a cause of the shrimping industry decline? Well, the shrimping industry in Georgia has been declining since the mid to late 90s. And this corresponds with the discoverance and emergence of blackgill along the Georgia coast. But we all know that correlation doesn't imply causation, and there's many challenges facing the shrimp industry, including like overseas farming of shrimp, as well as like boats that are in need of repair, and a younger generation who isn't as interested in participating in the shrimping industry as the previous generation was. But there is a strong correlation between blackgill prevalence and declining shrimp landings, particularly for the fall white shrimp season, which is the one that has the highest peaks of blackgill prevalence, as well as the one that's the most valuable for the shrimp industry. Also, importantly, there's a strong public perception that shrimp blackgill is affecting the shrimp industry. There's a big public outreach component to this project, and we go on blackgill cruises, as we call them, where we discuss our research with local shrimpers, and they reiterate to us that like the season goes strong until the mid to late fall, where the shrimp just kind of disappear. Additionally, the bait shrimp industry has been hit hard by this disease because of the damage that the melanizing tissue does to the shrimp gills. Shrimp are dying a lot faster in the holding tanks, really harming the shrimp bait industry. Okay, well, thank you so much for listening to my presentation. And thank you to all these individuals who have given me so much help over this past couple of years, and will certainly continue to help me during the next 3 or so. And then thank you to my funding sources.
Awesome, great talk, Megan. Very interesting topic. And we've got plenty of time for questions now. If anybody has any for Megan or suggestions for her as she moves along in her plan.
Hey Megan, this is Jan. That was a really nice talk, interesting pathogen that you're working with there. It looks like that this is a pretty noticeable parasite. It causes black gills. When was it first detected? I noticed that paper you cited was 2020 when that was first described. So it seems like this wouldn't be missed that easily since the gills turn black on there. Is this something that you're just seeing all of a sudden right now? And do you think that maybe it was introduced from somewhere, or why are you seeing these infections all of a sudden now?
Yeah, that's a great question. So it was discovered in 1996, but only recently has been reaching like really high prevalence. It is super noticeable, and we are actually working on a black gill tracker app for local shrimpers as well as people who just like shrimp recreationally to try and like track the spread of the visual black gill. It's difficult to determine as of right now whether it was introduced. There's a lot of correlations, like there's an invasive porcelain crab species that has come into coastal Georgia that could have been a potential vector, but we're really not sure. There is a strong connection with climate though, and we think that maybe if temperature is shown to be a major driver of transmission, like the increasingly warmer waters that we're having off the coast of Georgia could be why we're seeing suddenly this like high prevalence. So we've known about shrimp black gill since 1996, but it's only recently has it become a problem and a recent research priority.
Okay, thanks. And it didn't look like salinity has as much of an effect, right? Because from what I understand, this looks like it's an ectoparasite on the gills. So you would think that maybe salinity changes, like other, for example, fish ciliates are sensitive to salinity changes, and often that's a treatment. So what are your observations? I think you did mention in one of your slides that that doesn't seem to be as big of a factor.
Yeah, we haven't formally evaluated salinity changes. I do hope to address some of that in this like 3 habitat deployment where the marsh has lower salinities, estuary kind of medium leaning towards high, and then the ocean being like fully saline environments. Though of course it'll be hard to untangle the abiotic versus biotic components to any changes in transmission that we see across the habitats, but it might point us in a like salinity kind of direction if we do see differences.
Sure. And is there, are you trying to get this into a lab? Like, do you know, it kind of reminds me of ick, white spot disease, where you have the external stages, like the trophont, tomont. There's probably some incubation period where it needs to be in the sediment before tomites are released and are infected to the shrimp. So are there challenges with trying to purify this, getting into a laboratory and see if you can directly transmit shrimp?
Yes, we have not actually observed the swimming tomite stage. I tried like a little mini experiment where I had exposed shrimp in like 2 liters of water and filtered the water. And we were able to detect the like H-liney DNA from that. But I don't know if it's just like environmental DNA or whether it was actually the tomites. I want to talk to Dr. Landers, who is the one who like did this diagram and has done a lot of the morphological work for us to see if we can actually identify the tomites. But I'm hesitant to look under the microscope because I don't know what it looks like. It'll be like trying to find a needle in a haystack and not really knowing what the needle looks like. So that's something that we would love to be able to do, is to actually have like an ability to dose tomites to shrimp. But as of yet, we haven't been able to reach that capacity. We have looked at the seawater and haven't found a positive, like haven't been able to detect DNA from raw seawater or sediments. So that's part of the component too. But again, it's like the whole ocean, so it's hard to like say whether the small amount that we're able to sample is like really representative of what's going on.
Sure, that's interesting. Thank you.
Thank you.
Hi, Megan. This is Gail Kirath. I don't know if can you guys hear me?
Yes.
Yes, thank you.
Wonderful talk, really interesting stuff. I especially like the experimental design on the frequency versus density dependent transmission. That's a really important question for all infectious disease, and there aren't very many systems that have been well designed to study that. My question comes from, we've done a little tiny bit of it with IHN virus, and when you are trying to design the experiment to have various densities, there's two ways you can change the density. You can have different numbers of animals in the same volume of water, or you can change the volume of water. You can change. So I guess there's two ways to design this, and I'm wondering which you're doing in order to create your density conditions. Are those all in the same volume of water?
Yes, I think I am. I've debated that as well, because a concern that I have is like the water quality differences, because I'm trying to measure like individual contact rate differences, but I also don't want to affect like the water quality and have like the shrimp in the most dense treatment end up getting infected just because they're the most stressed. So that is a concern that I have. I'm hoping that like I'll just have as large enough volume of water that the highest density treatment is like perfectly happy, and I'll change the water a lot and frequently, so that doesn't become as much of an artifact. But that's the approach that we're going to go with, I think.
Yeah, that's we ended up with that sort of lucky situation. But if you can imagine if you have 8 shrimp in either half a liter or 2 liters or 4 liters, but it's the same number of animals, just a different volume, because having a different number of animals could impact the transmission as well. Not just, so it's really, it's a tough question. I don't know the right answer, but I just was curious. I figured you had thought about it, but it's going to be very.
Yeah, definitely. That is something that I'm like still struggling with a little bit. We haven't yet tried this experimental design, but I think like since we're trying to specifically test density, that I am going to keep it the same body of like volume of water across treatments.
I don't think there's any right or wrong. I think it's important to be able to think about, what each one gives you. And I think your design looks great. It's going to be very interesting.
Cool. Thank you. I appreciate that.
Great. Is there any other questions out there? All right. Megan, not hearing any. Thank you very much for presenting today. We really appreciate it. Virtual